Welcome to use full-quant SF real-time quantitative PCR system. The following video is a brief tutorial of full-quant SF analyzer software, which helping you quickly start to use the full-quant SF.
Start the software and create a new experiment. Click Experiment Setup directly, which at the bottom right of the interface, or select File, New Experiment and Experiment Setup from the menu bar, or click from the template to create a new experiment based on the saved experiment running template. Click Experiment Setup here. A new experiment setup process is as follows. Experiment property setting. Experiment name. The default is the current time, which can be customized. Tube type. Transparent tube is the only option. Type of experiment, including standard curve, comparative CT, double delta CT, genotyping. Here we take SARS-CoV-2 detection as an example for the following instruction. It can be done with the experiment type of standard curve, thus the standard curve is selected here. Chemistry. The TAC-main reagent and sub-green reagents are optional. SARS-CoV-2 detection commonly use TAC-main reagent as an example here. After setup is completed, click Next in the lower right corner of the page or the method on the left to get into the running procedure setting interface. The default reaction volume is 20 microliter, which can be customized, and the thermal cover temperature is 110 degrees Celsius. Reaction stage setting. Set the reaction stage and steps according to the actual reagent instructions. If you need to increase or decrease the PCR reaction stage, such as installation PCR reaction, etc., you can move the mouse to the reaction stage area and click Add or Subtract button. The Add button on the left and right means adding another one before or after this stage, and the Subtract button in the middle means removing this stage. If you want to add or remove the reaction step in a certain step of PCR, you can move the mouse over the step area, click on Add or Subtract button. The left and right Add button represent a step before or after this step. The middle Subtract means to remove this step. The reaction of temperature, time, and the number of circles can be increased or decreased by inputting numerical values in the numerical values area, or clicking the up-down buttons. The temperature setting can also be changed by clicking and dragging the temperature curve. The camera icon same as the signal acquisition icon. When the icon is dark blue, it means that signal acquisition is on, and when it is light blue, it means that signal acquisition is off. The software defaults to signal acquisition after each cycles of application. Getting into the plate setup after setting is completed. The plate setting can carry it out before, during, or after of the reaction. In order to save your experiment time, it is recommended to set it after the running reaction. Taking the SARS-CoV-2 nucleic acid detection as an example, detection of the SARS-CoV-2 ORF1ab gene and the nucleic acid protein engine using fluorescent probe. The specific settings are as follows. Click Advanced Setting to add samples and the target genes. Click Add to add samples as well as the target genes. You can also rename them. For example here, name the sample as NTA positive, unknown sample 1 and unknown sample 2. Name the target 1 as ORF1ab gene and select FAM as reporter channel. Name the target 2 and N and select week as reporter channel. Next, you need to assign the information of samples and target genes, which has been set to the related well positions according to the actual sample's placement position on the left well setting interface. Drag the left mouse button to select the related wells and then check the corresponding sample name and target gene on the right advanced setting interface. Click Start to run. The system will pop up a data saving dialog box. select the saving path, click the saving button, save the file to the select path. The default file format is qPS. After saving, the system will start to run the experiment automatically. You can see the real-time temperature information of the thermal cover and the thermal block on the upper left corner of the software interface, as well as the remaining time of the current experiment at the upper right corner of the interface. When the running procedure is completed, the result will be automatically present in the analysis interface. Here we take previous SARS-CoV-2 detection results as an example to illustrate the process of result analysis. Select all samples, click Analyze. You can view different types of result plots through the drop-down menu in the upper right corner. Here, we select the amplification plot. The amplification curve will be shown up, and then you can review the all-over amplification result of different samples and target. Take a SARS-CoV-2 detection result as an example. Normally, the sample should be determined as negative when its CT value greater than or equal to Y or with no value. And it should be determined as positive when the CT value less than or equal to X with classic S-type curve. For the practical application, you should refer to the instruction of the detection reagent kit that you use to judge the results. When viewing the amplification plot, you can click a point on the curve, click the Zoom In button to zoom in, and click Zoom Out button to zoom out. You can also send the current amplification plot to the printer for printing, copy the clipboard, or save as a picture. Click the Map Feature button to save at the curve title, X-axis and Y-axis. Click the Map Legend button to hide the map legend, and click again to restore. Click the Curve Setting button to set the plot type, graph type, target, threshold, etc. Click the list icon. The software will display the experimental results in a list, sample name, CT, and other experimental information. Click View to check or remove the types of the items that needs to be displayed in a list. Click the Well Played icon to return to the Reaction Well Played interface. After the data analysis is completed, enter the Result Report interface. File name. The default is the current experiment time, which can be customized. Content. The exported content can be selected according to needs. The system merges the above information into one file by default. File type. The file export format includes TST and XLS types. Choose XLS types usually. Location. The system default is the desktop. You can customize other paths to save. After the setting completed, click Export button. The results will be displayed in an open Excel. The table contains the planned settings, amplification data, multi-component data, results, and other sheets.